my talk. My talk today is about ion mobility mass spectrometry for structural characterization from a fundamental perspective. Basically, this seminar is all about material science, but today I am presenting more about a technique, an analytical technique, and probably you are thinking, what is the importance of this technique in material science? That you will come to know in the next slides. In today's talk, I will present about, you will learn about what is mass spectrometry and what is ion mobility spectrometry, basic types of IMS systems, that's ion mobility spectrometers, and then I'll discuss about trapped ion mobility mass spectrometer. Finally, I will conclude by explaining how we can measure collisional cross-section with a TIMS device. So today's talk I will take in a pedagogic way because I have a wide audience. Most of the audience are not specifically working in mass spectrometry. Probably, maybe some of you are familiar with the mass spectrometry, but I'm sure only a few people probably may be working on ion mobility, because ion mobility is a very recently developed field, but it is fast growing and having very high application, very high demand, and is a very hot field. So as this slide shows, ion mobility mass spectrometry has a lot of applications. For example, this slide just shows a wide area of application, not specifically, but if I start saying all the applications specifically, it take a long. So I just summarize all the things in this slide. It has application in pharmaceutical, medical diagnosis, diagnostics, clinical, and obviously material science. I just pointed some of the applications in material science because this seminar is about material science. And in material science, we use mass spectrometry to identify the purity of the materials, to study the polymers, surface analysis, to identify the chemical structure, and also for the chemical imaging of the surfaces, and also the interface studies, more and more. And also this has application in national security, airports, seaport, detecting warfare agents. Most of the time, when you travel through airport, probably you might have seen the security come with a small tissue and wipe on your hands and on your luggage, and then take in some device and then come back and say, you can go. Actually, what they did, they took the tissue to a ion mobility mass spectrometer, and they put in that, and then they identify if there is some sort of explosives or something you handled for last a few days. Even if you handled 2 days ago some sort of explosives, the trace amount will always remain on your hands. However you wash, it will still be on your hands. They still detect it. If there is some sort of explosives or some kind of narcotics, some drugs in your bag, the trace amount will be somewhere left on your bag. That's what they are going to do. They are going to identify. That is how ion mobility is used in national security, and also environmental to detect the environmental pollution, forensic examinations, drug detection. So what they do is, like, if some sort of crime happened around you, then the forensic offices, they come collect the samples and they send to the forensic labs. There are a lot of kind of analytical or several kind of experiments they perform there. One of the top, highly sensitive technique is mass spectrometry, especially ion mobility mass spectrometry. It can really and very perfectly determine what kind of chemicals and what kind of things are on the samples, and they can use it for the forensic identification. Another application is proteomics analysis. I am mainly working on proteomics analysis and also on national security applications. In proteomics analysis, to identify the proteins and to study the post-translation modification of the proteins. and mobility mass spectrometry is used. And here I pointed out some of the medical applications to detect the cancer, pulmonary disease, potential rejections after lung transplantation, breath analysis, like that, a lot of applications for ion mobility mass spectrometry. Now probably you understood what is the importance of ion mobility mass spectrometry. Today I will present you from the very fundamental perspective, or the how the ion mobility mass spectrometer works, and what are the kind of ion mobility spectrometer and mass spectrometers we are using, and then I introduce you about our Bruker Teams device. So let us begin. Before we start from the what we learned from the high school. In the high school, we learn Newton's law. Force is equal to mass into acceleration. What happens when acceleration is equal to 0? Velocity is a constant. And what does it mean? If we have different masses, and if we force or give energy to these masses and allow them to fly through some distance, what happens after some distance? The masses will arrive. Different masses will arrive at different time, because different mass acquire different velocity. That's the basic principle of mass spectrometry. We separate the mass according to the time of flight. So here, this shows here we have 4 different masses. These are ions. In mass spectrometry, what we use is we use ions, not just neutrals. 4 different mass ions. Okay, we accelerate these 4 ions in this region, in this region where we apply electric field, and this region does not have any field. This is a field-free region, and this is the detector. When we accelerate, different masses acquire different velocity, and different masses arrive at the detector at different times. That way, we can separate, or we can identify what are the masses, what kind of what mass molecules are in our samples. So probably you understood. Mass spectrometry is about separating the molecules or ions according to their mass to charge ratio, because it is in the electric field, and also the ions have charges. So these ions are not just separated according to their mass; they are separated according to mass to charge ratio. So this is called mass spectrometry. So the mass spectrometry is performed in vacuum. There is no air in this. It's an ultra high vacuum, 10 power -8 or -9 torr. If you see a mass spectrometer, it has several sections: an inlet, an ion source to produce the ions, whatever the sample you introduce, for example, liquid sample or gaseous sample or solid sample, whatever the sample, you can introduce the sample into the ion source and produce the ions based on several techniques: electron ionization, chemical ionization, means matrix assisted laser disruption ionization. This is laser ablation technique. If it is a solid sample, you can do laser ablation, or SIMS, secondary mass spectrometry. There are several types of ionization techniques. You produce the ion, that is the first stage of mass spectrometry, and then introduce the ions into the mass analyzer. This is the core analyzer I was talking about. This is the mass spectrometer. Then after the mass spectrometer, we have detector. From the detector, we have data storage devices where we store the data and do the further analysis. Although I explained you mass spectrometry based on a time of flight device, there are several types of mass spectrometers. We can separate ions according to mass to charge based on time of flight, and also based on different techniques. One is, here I show fundamental types of mass spectrometers. There are more types of mass spectrometers, but these are the types of mass spectrometers which are widely used and more fundamental. And I also mention here who are the industries or vendors of these instruments. Magnetic sector it uses just a magnetic field to separate the ions. This is used mostly in the academic field, and this doesn't have much resolution, but still, for example, if you have carbon clusters, you want to separate the carbon clusters, then you can use a magnetic sector mass spectrometer to separate the carbon clusters, because the carbon clusters are not much complicated. If you have a protein or biological samples, they are too complicated, way more complicated. Then you need a more highly accurate, more specific, more resolving power mass spectrometer. In that case, you have to use more advanced mass spectrometers. Another one is time of flight that I explained in the previous slide. Time of flight mass spectrometer, it just accelerate the ions using an electric field and then allow to fly through a field-free region. Time of flight is also modified into several types of, like reflectron and several different types. But in general, we can say time of flight. Main vendors of the time of flight mass spectrometer are Agilent, Bruker, SciEx, and there are more, but these are the major companies who work on time of flight mass spectrometer. Another one is quadrupole. In quadrupole, we have, as the name indicates, it has 4 poles. And in the 4 poles, we apply RF voltages alternatively out of phase. On 180-degree out of phase. These two has RF, and these two have another RF with 180-degree out of phase. These are called mass analyzers. This can be trapped also in a different shape, but we are not going into the details. You just understand this quadrupole analyzer can filter out masses according to mass to charge ratio, depending upon the amplitude and also the DC field we apply. It's a bit complicated, but I just want to introduce you. This type of mass spectrometer is produced by Agilent, Bruker, SciEx, and Thermo. There are more companies making, but these are the big giants in the field. Another type of mass spectrometer is more interesting one. It is orbit trap mass spectrometer. This is even invented by Professor Dr. Alexander Makarov, who is in Thermo Fisher Scientific. Obviously, this is made by Thermo Fisher Scientific. This has a special type of electric field, and this trap the ions within this field. The field model is given here. Here, the ions circulate around this rod and oscillate up and down. The oscillation frequency depends upon mass to charge ratio. Then they take the induced current from this metal surface and use the Fourier transform technique to convert into the real mass to charge ratio. This is a kind of trap device. This is not the only trap device in the field, but there are other trap mass spectrometers like electrostatic ion beam trap, which is developed in Weizmann Institute of Science. But those are not used for the analytical purpose much. This is the one which is used for the mostly for the analysis purpose. Other mass spectrometers are also used for depending upon what we want, what kind of analysis what we want to do. Okay. So these are the fundamental type of mass spectrometers now people are working on. Now let's go to the ion mobility. What is ion mobility? So you understood that mass spectrometer separate ions according to their mass to charge ratio, and it is operated in ultra high vacuum. But the mass to charge ratio is not everything about the molecules. This mass spectrometer is a kind of weighing device. It weighs the molecules. It tells us what mass the molecules are. And it can happen that 2 molecules have same mass but different shape. The shape is also very important. For example, especially in biological samples, or we can. I can give an example that, for example, in our eyes, in our eye lens, how the cataract form. For example, if the eye lens is exposed to, if our eye is exposed to UV light, or for example for some other reason, because of our food or for some reason, if the proteins in our eye lens change the conformer shape, change the shape, then the eye lens become opaque, right? The protein, if the protein stay in some shape, it has some specific property. If the proteins change the shape, it loses the property. Another example is Alzheimer's disease. In our brain, in specific areas, if the proteins change the shape, it lost the capability to store the memory. So the shape is very important. And also about our skin, when we get a lot of sunlight, the proteins on our body change the shape, and it induces the production of more melanin, and we become tanned, right? So these are some of the examples where the shape of the molecules are very important. So it is not all about the mass; it's about the shape too. We get the shape information from IMS. IMS is the technique where we identify molecules based on the shape. So most of all the researchers in the world, most of the researchers in the world, how they work is they use ion mobility spectrometer in conjugation with mass spectrometer. They initially separate the ions according to their shape, and then introduce into the mass spectrometer, and then they do the analysis. How they do the analysis, we will see in the next slides. So this is how most of the research teams are working on, but standalone devices are also possible. Ion mobility standalone device is possible. For example, in the airport, the detection device is a standalone ion mobility device. It doesn't have a mass spectrometer. And there are a lot of mass spectrometer. Mass spectrometer can also be standalone. Most of the mass spectrometers till now were standalone, and for last 10 years only, the ion mobility spectrometer become that much demanding. So here in this slide, I am going to show you the difference between the fundamental difference between mass spectrometer and mobility spectrometer. Let's assume an experiment. Take a full-size paper, two papers, and one paper as such, another paper crumble, like it is shown here. And if there is no air, if it is in vacuum, what happens if you drop from the surface from a height? Both fall the ground at the same time. If we have a detector here, the detector detects both the masses at the same time. This doesn't, the detector doesn't know which one is crumbled and which one is the non-crumbled normal sheet. If you do the same experiment in air, what happens? The crumbled paper will fall down first, and the other paper will fall down later. If we have a detector on the floor, the detector can easily say, hi, this is the crumbled paper and this is the unfolded paper. This is ion mobility spectrometry. In ion mobility spectrometry, we introduce some background gas into the chamber so that the ions separate according to their shape also. Here, this is a gravitational analogy, but we do with electric field. So there will be more parameters depending on the charge and polarity and all and all. But basically, this is the difference. Mass spectrometry we do in vacuum, ultra high vacuum, but ion mobility spectrometry we do in the presence of a background gas. And mass spectrometer give you the information about mass discharge. At the same time, ion mobility spectrometer give you information about the shape of the molecule. Scientifically speaking, it is better to say collisional cross-section. The shape, the information about the shape is in the collisional cross-section. Means, I'll tell you what is collisional cross-section, how you can imagine collisional cross-section. Probably you already studied about collisional cross-section in physics courses. Here in this slide, there are two molecules, two proteins with the same mass but different shape. These two have two different collisional cross-sections. This is how you can imagine you can visualize a collisional cross-section. In your textbooks, you studied collisional cross-sections like what I show in the picture on the right side here. The area of the cross-section, you define the area of the cross-section as the collisional cross-section. But you should visualize in this way. If you have a background gas in it, the probability of the molecule to collide with the other molecule. This is the collisional cross-section is a kind of surface. It's a dimensional surface. Here we study it's like an area, but when we visualize, we should visualize in the form of a surface. Yeah. Okay. Now let's go to ion mobility. If we have a background gas, as I explained, these blue balls are the background gas. This represents the background gas, and we have ions. Ions are represented by red dots. We have ions here, and if we apply an electric field, what happened? The ions started migrating towards this direction. it is negative charge, it will move in opposite side. And the motion and the speed of the ions depend upon the pressure inside the chamber. Right? We can apply the binary collision model and we can derive the velocity to be Vd, which is called drift velocity, is equal to Ke. E is the strength of the electric field, and K is called mobility. Mobility is expressed by this equation, and in this equation it has omega, which is called collisional cross section. But again, very careful here. Omega is called collisional cross section, and we all in air mobility field we all call it collisional cross section, but it is not exactly collisional cross section. It is in fact the momentum transfer collisional integral. The momentum transfer between ions and particle average over all the gas ion relative thermal velocities. That is here. So it depends upon the how polar the molecules are, and the dipole moment of the molecules and all. But still, this has the information about the cross section. This is what we want. If we get this information, we know the shape of the molecule, and the shape of the molecule. And here the mobility, we get the mobility in this particular pressure and temperature. So when we somebody do the experiment in some pressure and temperature, and another person in another lab doing another pressure and temperature, we can compare it, right? So we have to compare it. So we always talk the mobility in terms of reduced mobility. We convert it into standard format, and we talk about reduced mobility always. Yeah, once again, you can imagine visualize this in the form of, for example, you assume a room having a lot of people inside, and then a fatty person and a slim person try to go through the room. The slim person, he will easily go through the room because he can easily go through. How? Because he has less size, right? The size is smaller. This we can say that the collisional cross section is smaller. So if the molecule is bigger, it has higher collisional cross section. If the molecule is smaller, it has smaller collisional cross section. It will be easily passing through the background gas. So the smaller molecules means molecules which are, for example, if you assume the normal paper and crumpled paper has smaller collisional cross section. That means higher mobility. It has higher mobility. It can easily move through the background gas. The normal paper has higher collisional cross section, so it has lower mobility. So we can measure the mobility. If we get the mobility, we can say about the collisional cross section. This is how the experiment goes. Here it shows another demonstration how the molecules are separated in the background gas and how we do the measurement. Here we have two molecules. One is both are same mass. As in these are proteins. Native protein means protein in the same shape as such how it is appear in the biological systems, and denatured proteins means proteins which are stretched. You have same protein with one on the native form, on the stretched form. Both have same mass, and it passes through the chamber, and the native one reached first and the other one reached later. This way we can separate. If we have a detector, the detector will give signals at two different times, and the one arrived later time is the one which is denatured. That way we can separate we can identify what kind of proteins these are. What I explained up to now is the very fundamental form of ion mobility. There's a very basic form of ion mobility, and drift tube is the best example for such an ion mobility device. It's very simple. It has an ion source and then a chamber with a gas filled in it. Gas can move or can be static, and then ions are allowed to pass through the background gas by applying a DC field. In this case, this is exactly like what I explained previously. This kind of ion mobility spectrometers are commercialized by Agilent Technologies. Basically, this IMS and MS, this ion mobility spectrometer coupled with the mass spectrometer was developed in Pacific Northwest National Laboratory, and then Agilent took the license and they started working on it. They continued working on it. And now they are the one who are the vendors of the official vendors of these kind of spectrometers. This is very simple, and the researchers can directly plug in the equations from the theory. Well, if you look at the theory, there are a lot of theory. Many people work on the theory with how the ions move through a background gas when there is a uniform electric field. In this device, you can plug in all the equations right away, and you can calculate the collision cross section. It is a very simple device. This is very easy to work on. But there are negatives that I will show you later. The type of mass spectrometer is developed by Waters Corporation. What they did was they removed the DC field and they applied a traveling wave like this. The wave is traveling. The problem with the DC field was that if you have higher resolution, if you want to separate the ions better, you should have longer drift tube. When the drift tube is longer, you will have to apply higher voltages. Higher voltages means you will have to apply kilovolts, very, very high volt, which is dangerous, and sometimes it is not practical too. There are problems again. If the device is that long, the ions will stay in the device for a longer time, and ions start taking energy from the electric field, and it's changing the size and shape. Then there is no point in analyzing in this sample, right? If the ions keep on changing the shape, then it's impossible to analyze the device. So here Waters invented another kind of device with a traveling wave. Here the advantage is that you don't need to apply very high voltages, and the resolution is even better because the mobility and velocity relation is a square dependent, and they have the theory. You can find the theory on about the traveling wave on literature search very easily. This kind of devices, it has a better resolution with the same length, and you need to use only very low voltages, 10 to 50 volt voltages. It is based on how the ions travel through this traveling wave. The ions in this device will be taken forward by the traveling wave, and while moving, the ions will roll back. The number of rolling back depend upon the size and shape of the molecules. That's how it is separated. I'm not going into details of the traveling wave, but I will go to another type of mass spectrometer, which is developed by Bruker. In 2010, Bruker came up with the first publication about the new kind of ion mobility separation device by applying a kind of gas velocity and also DC field. This is the first publication about a new kind of device from Bruker. You can find it in the literature search. How it works? What is the specialty of the device? This device has a gas flow in the forward direction and electric field in the backward direction. When the gas tries to take the ions in the forward direction, the electric field will oppose the ions. The gas will take all the ions independent of their mobility in this direction, but the electric field tries to separate them in the opposite direction. We'll see how it is. So it has two phases. First, we accumulate the ions in this region, and in the accumulation phase, the ions stay in this region. This plot shows the electric field in this device. The ions stay in some position based on the velocity of the ion in the gas and the velocity due to the electric field. If the velocity of the ions due to the gas is in forward direction, and at the same time velocity due to the electric field is in the reverse direction. So ions will stay in these positions depending upon at which position each ion, the velocity of gas plus velocity due to drift. Drift means velocity due to the electric field. Okay, this velocity due to the gas, and this is the velocity due to electric field. This velocity, the sum becomes zero. There, the ions stop. So this velocity due to drift depend on mobility. So because of this relation, the ions with a lower mobility will stay up the ramp, and with the higher mobility will stay down the ramp. Then once the ions stay in this position, accumulate in this position, then the next phase starts. Then we start eluting. Then we change the field a little bit. Then the first ion elute. The other ions change the position and again stay stationary in another position. Then again we change the field. Then again change the field this way. This way we have very high resolution, and we can identify the molecules based on their size and shape. This is called trapped ion mobility mass spectrometer. This is developed by Bruker. And how it look like? This is the cross section of the real TIMS cartridge. It has a funnel, and it has the TIMS analysis region, and then exit funnel. This is the ion optics to take out the. This is the first spectrum Bruker got in 2010, and this is in 2016. We improved a lot. We changed a lot. We made a lot of modifications to improve the device. And now we have super good resolution. This is the time history of the development of Bruker TIMS device. In 2011, it came up with the real the publication product. It was published by Melvin A. Park in Bruker, Billerica in USA. And then next publication was from Matthias Mann lab in Max Planck. Bruker collaborated with Max Planck, and we published another paper. There are a lot of papers in between, but these are the key publications. In 2016, we launched the timsTOF device, and then we came up with the timsTOF Pro with some modification. In 2018, we made further modifications, and 2019 we launched timsTOF Flex with a lot. And you can see the publication. This was again published with Matthias Mann. What is timsTOF Flex? timsTOF Flex has a dual ion source with a multi. Or the ion source, you can choose whatever the ion source you want. If you have a liquid sample, you can choose electrospray or whatever the ion source. It can be modified. This is the TIMS device up to here, and then we have the mass spectrometer. We have quadrupole mass spectrometer and time of flight mass spectrometer. Here you will see what is the use of all of the use of quadrupole and time of flight mass spectrometer. This is the part we have the TIMS analysis. This is the ion mobility part of the device. We introduce the samples through this way and do the ion mobility separation. And after choosing which kind of molecule we want, then we allow the ions to go through here, and we can do a lot of analysis that I will show in the next slide. One of the application, if I show you quickly, it is, for example, if you have proteins. Proteins can be normally how the proteins are identified is called bottom-up approach. Means we take the proteins and then digest it and then introduce into the mass spectrometer. In that case, what happens is that the proteins already lose a lot of information. In this case, what we can do is we can introduce the protein as such, and then take the isolate the protein, and then break the proteins in this region. Then do the analysis. So this is more efficient approach. So here I am showing you a bit more advanced timsTOF system, the passive MS scan. What is passive? Passive means this letters stands for parallel accumulation sequential fragmentation. Parallel accumulation means this is while the spectrometer do the analysis, the TIMS keep on accumulating the sample. That's parallel accumulation. That what's the advantage? The advantage is that it is 100% duty cycle. The TIMS is not sitting ideal. I mean, it's not doing doing anything during the analysis. It keep on doing the job always. At the time the mass spectrometer do the analysis, the team staff keep on collecting the samples. So it has a 100% duty cycle. How it work? We introduce the ions here in the team cell, and then this is the accumulation region, and then this is the TIMS analysis region. We separate to the TIMS region, and then we allow the ions to pass through the mass spectrometer. In the mass spectrometer, it's separate according to mass to charge ratio, and we record the data here. So we know what are the kind of molecules we have. For example, we can talk based on the proteins. Okay, for example, if you have a tissue or if you have a biomaterial, any biomaterial with proteins, and you want to identify proteins. Okay. And then the next stage, we select those proteins and then fragment each protein, and we record the fragments of each protein. That way we have all the information about the protein. This is we have the fragments, then we assemble it together and we understand what it is. If we have these fragments, we have the full details, the whole details about the molecule. We know about the fragments, what are the components in the proteins, what is the post-translational modification, if there is any modification. For example, if you take a sample from a normal patient and normal person and disease affected, for example, some sort of, for example, Alzheimer's caused patient, Alzheimer's disease affected person, and from a normal person, if you take the brain tissue and if you try to identify the proteins, you can identify the difference. You can find the protein, and also from these fragments you can identify what kind of modification it happened. That's the beauty of this. This is how it looked like. The reality. You have you get the heat map like this. Then the heat map, from the heat map, three-dimensional heat map, you can identify the samples. This is the MS spectra, and this is MS heat map. Then from here you can identify each fragments, the molecules and the molecule fragments. Molecule, when I say molecules, it's for the big molecule, proteins and the fragments. So, now you understood what is the interest, or what is the one of the applications. There are a lot of applications, but I just show you one application among a huge lot of applications. Now let's go further deep into the analytical model of the trapped ion mobility mass spectrometer. This was the one of the theory paper published by Bruker about this TIMS device. You can find this paper. There are two parts for this paper, part 1 and part 2. You can find this, and there are a lot of other theory paper came out, but this is the core theory paper published. I will show you some of the very important results from the theory. Here we are comparing the trapped IMS with the standard drift tube. Okay. In the standard drift tube, the resolution is given by this expression. And trapped IMS also, the expression came to be the same expression, but with a difference. The L, the length. Here Q is the charge, E is the electric field, and Boltzmann constant temperature, and L is the length of the device. In the drift tube, L is the physical length of the device. But in our case, L is the effective length, which is given by VGTP. VG is the velocity of the gas, and TP is the time ions spent in the Plateau region, in this region of the electric field. So it is not about the physical length. What is the advantage? In the drift tube IMS, if we have 2 meter length, for example, assume the 2 meter long, to have some specific resolution to identify some molecules, we need 2 meter length drift tube. We can have 10 meter length with this small physical length device. VGTP. How? By increasing the velocity of the gas. We are not increasing the physical length. We are just changing the velocity of the gas, and we are attaining equivalent to length equivalent to 10 meter. With this 2 meter, they get 100 resolution, we get 200 resolution. To get 200 resolution, the drift tube has to be 10 meter long. Assume a 10 meter long device sitting in a place. It occupies the whole building, it has the two or three rooms. And then you will have to do analysis. And this takes a lot of space, expensive, and also you have to apply very high voltages, which is again dangerous. And also other problems like for 10 meter long, if the ions spent in the electric field, obviously ions are going to change the structure and shape. Then you are not going to get the information, right information. But here, within 0.05 meter, this much length, you get 10 meter equivalent length and get resolution of 200. Another problem, if you look at the Wikipedia about the drift tube, you can see that the collisional cross-section equation is given by Mason-Champ equation. What Agilent, the company Agilent claim is that they can get the information about the collision cross-section directly plugging the results into Mason-Champ equation. And they say that Bruker cannot give you collision cross-section by directly plugging in, but that is wrong. We developed another theory that we can directly calculate the collision cross-section from the device. But it is true that drift tube is very simple. The drift tube, we can plug in the equations directly without any modification because a lot of people work on this theory, and there's no more complications. You can easily plug in the equation, the results into the equation, and you can find the collision cross-section or whatever information you want. But the Bruker teams also, we developed another theory which we can plug in the results directly, and we can calculate the collision cross-section. That is going to explain here. I'm comparing drift tube and trapped IMS results here. The reduced mobility is given by this equation in the drift tube case, and in the trapped ion mobility case, the reduced mobility is given by this equation. These two equations are the details how it is derived is given in the theoretical paper that I shown in the previous slide. If you download the theoretical paper, you can see all the derivations of. I don't want to spend time on deriving this equation here. Just go to the result and try to explain the result. So this is the mobility expression for the drift tube. And in our case, the reduced mobility is given by this equation. Here, the Vg means velocity of the gas, and Lp means length of the plateau region. The electric field has a plateau region here, the length of the plateau region, and T is the time of elution. Tt is the transfer time to the mass spectrometer, and beta is the scan rate. We scan this electric field right from high to lower value. The scan speed, beta is the rate at which the electric field is changing, and this is the factor to transform the mobility we measured here to the standard mobility. From this equation, the derivation of equation is somewhat easy, not so easy, but you can see that in the theoretical paper. If we take that equation and if we rearrange that equation so that we get the elution time, if we take out the elution time here and do the rearrangement, you can easily arrive to this equation. Here, beta is the one we scan. This is the dependent parameter. We change the beta, and then we take out the sample, right? If we take beta, 1 by beta, square root of 1 by beta as x, then we can express the equation in this form, 1 by beta in the x, and then here also x square, here also x square, and take the common term out of the x square, then we can get this one, and here x and tt. This equation is very simply we can rearrange. This is a second degree polynomial. We have a second degree polynomial. We have a and b here. If we have the result of the experiment in time of elution of the sample, we can fit into this second degree polynomial easily. If fit into the second degree polynomial, then we can easily tell, we can, from the fit equation, we can find out what is A and what is B. If we have A and B, then we can easily find out K. If we have K, we can easily find out K0. This is just about fitting the results and do some calculation. That way we directly calculate the correlation cross section. We don't need any other calibrants. So what Agilent claimed was that Bruker needs some calibrant solution, some calibrants, or need to know some information about prior information about collision cross section in order to calculate further the collision cross section of the other molecules. But that is not true. We can easily plug in the equation results into this equation, and we can directly calculate the collision cross section without any calibrants. So this is the comparison between drift tube and other kind of mass spectrometers compared with the trapped IMS, TIMS device by Bruker. Drift tube, only drift tube, up to now, drift tube could give you the collision cross section information directly from the without any calibration. Direct measurement of collision cross section was possible only with the drift tube, but now direct measurement of collision cross section is also possible with the, and TIMS has much more capabilities than a drift tube also. In summary, what we learned today? We learned today about IMS and MS basics. IMS means ion mobility spectrometry, and MS is mass spectrometry. What is mass spectrometry? Mass spectrometry is the technique where we separate ions according to their mass to charge ratio, while IMS is a technique where we separate ions according to their size and shape. MS is done in ultra high vacuum. At the same time, IMS is done in the presence of a background gas. There are several types of MS: magnetic time of flight, quadrupole, and trap are the fundamental type, and much more type of mass spectrometers are available, but these are the fundamental types of mass spectrometers. And we have several types of drift tube ion mobility spectrometer, drift tube IMS, traveling wave IMS, trapped IMS, and there are more types of IMS systems, field asymmetric, structures for lossless ion manipulation devices developed by Pacific Northwest National Laboratory, and those are advanced techniques, but these are more, we can say, more fundamental or more widely used techniques. Structures for lossless ion manipulation, these are the devices just launched, and a new company called Mobilion, spin-off from Pacific Northwest National Laboratory, they're trying to commercialize it. Now they took the patent for the structures for lossless ion manipulation devices. Probably in coming years here, it will be more into the field. And Bruker has TIMS, which is a very powerful IMS system. We can do top-down proteomics, and also we can have direct measurement of collision cross-section directly from the TIMS system. These are the takeaway points. If you, although if you miss some of the parts from the presentation, I think at least you will remember at least these things. So about my presentation, thanks, and thank you very much.